Back. This is part 2 of cell-cell junctions. Last time we looked at cadherins and adherins as mediators of cell-cell interactions. So what we're going to look at here are non-cadherin mediators of cell adhesion and also cell recognition. So let's get going. So besides cadherins, there's a whole bunch of other molecules that are involved in cell-cell adhesion and in recognizing, in allowing cells to recognize one another. For example, many animal cells and plant cells, for that matter, secrete a carbohydrate binding protein called a lectin. So a good example of that here are the selectins, and so you can see lectin as part of the name selectin. A whole family of proteins, P-selectins, E-selectins, L-selectins, large family, and they promote cell-cell adhesion by binding to a specific sugar or sugars on another cell surface. And so they could bind to two carbohydrates on two different cells to bind those two cells together as well. So those are selectins. So that's a protein which binds to a carbohydrate on another cell. There's another group of molecules here which we touched on last time a little bit, the CAMs. They're members of the immunoglobulin superfamily, the IG superfamily. So you probably heard about immunoglobulins in terms of antibodies. The main member of that family is the neural cell adhesion molecule, or NCAM. They have these kind of loop, these well-organized loop structures, which are related to the structures of immunoglobulins, hence the name, hence them being part of the immunoglobulin superfamily. And so they can be involved in homophilic interactions between cell types. That's what we're going to talk about there. Let's look at an application of how selectins work, though. So if you've seen that video, The Inner Life of the Cell, I posted it a few times in the Zaptions. If you've not seen that, go to YouTube and look for Inner Life of the Cell. And in the first part of that movie, is what you see is a leukocyte circulating through vasculature, binding to the endothelial cells, which are the cells which form the inner lining of a blood vessel, and then the leukocyte extravasates out of the vasculature into the tissue. So when what we've got going on here is that you have a leukocyte which is kind of rolling and tumbling through the vasculature, and on the surface of leukocytes are L-selectins, L for leukocyte, and then we've got P-selectins and E-selectins on the surface of either endothelial cells, or in the case of P-selectins, platelets, but let's just think about E-selectins here. And so the L-selectin, what happens is cell surface carbohydrates on the leukocyte are going to engage with the P-selectin and E-selectins of the endothelial cells, and that kind of slows the tumbling of the cell down and initiates this weak association between the leukocyte and the endothelial cells. And it also triggers a bunch of signaling to activate the integrins, and the integrins associate with some members of the CAM family, ICAMs, which we just talked about, and that's a tight binding, and that tight binding between the leukocyte and the endothelial cell allows the leukocyte to extravasate out of the vasculature and into the tissue at the point of inflammation. So you want to see that happening, take a look at the Inner Life of the Cell video. That's all I'm going to talk about in this video. It's a nice short one. I'll be back next time with more cell-cell junctions stuff.